Hey everybody, so we're going to get into chapter 4 and look at the digestive and absorption process of our bodies. Let me share a screen here. Okay, you should be able to see, hopefully. And now. Okay, so chapter 4. So basically what we're going to do now is we talked earlier about energy expenditure and energy consumption. So when you eat those carbohydrates, those fats, and those proteins, what happens to it? So we're going to give you a rundown of the digestive system, and then we'll take it into absorption and where those and how those carbohydrates, proteins, and fats go from your intestinal tract into your body. And so remember, when we put things in our mouth, we're basically putting it inside of a tube inside of our body, right? So it'll go in one end and right out the other, as you can imagine. And somewhere in that process, we have to take the nutrients that we eat, whether it's energy or in a form of a carbohydrate, fat, or protein, or some other macronutrient or micronutrient. So whether it's a vitamin, a mineral, water, those kinds of things, they have to get out of that tube, that digestive tract, into our circulatory system so that they can be moved around our bodies. So that's what we're going to talk about. So basically, here is the systems that I want to talk about, the purple arrows. So we're going to talk about the mouth, the esophagus. We're going to then lead into the stomach, talk about the small intestine, and then from there, what the pancreas and the gallbladder does. And then, of course, the small and large intestine. So we'll move through these. This is just an overview. So we're going to hit each one as we go. So let's start in the mouth. Remember, this is the digestive process. So we're going to put food in our mouth, obviously. And the reason why we chew it, and it's called mastication, that's the mechanical process. We break down the food. We're trying to make it smaller. We're trying to make it flatter so that it has more surface area. And inside of our mouth, we're going to secrete some enzymes and begin some of the digestive process with enzymes. Most commonly, we're going to put enzymes that will break down carbohydrates first in our mouth. So those come from glands. Those enzymes come from glands. They're listed right there. And they're located obviously in your mouth. So as we chew, that's why we get the saliva that's mixing in with the food. The food then passes down our esophagus. Now our esophagus is lined with smooth muscle, and that smooth muscle performs a rhythmic contraction called peristalsis. And that peristalsis contraction requires energy. So if you remember back to the last lecture, that's where you get the DIT, right? Dietary induced thermogenesis. That's where the DIT comes from. So the smooth muscle contraction that's going to start in the esophagus and continue to move that food down through the stomach, into the small intestines, through the small intestine forever, and then into the large intestine. And then, of course, then we defecate. So we get rid of it. So all of that is done by peristalsis, begins in the esophagus. We do move it through a couple of sphincters, which are also muscle, part of that DIT. Those sphincters let food pass in through, but it doesn't let it come back up. A lot of times when we throw up, that esophageal sphincter will open up and allow food pass out of our stomach. And obviously that's not a common thing, right? Or it shouldn't be. Okay, so that's that. Now you'll have to forgive me on the stomach because there's a couple of things that I need to change on this slide and haven't gotten to it. So here's your picture of your stomach, and there's the listing of the esophagus. There's the sphincter. The first one is the fundus, which is correct over here. Oh, I did change it. Look at me. Oh, except I didn't spell it right. It's antrum, not atrium. I'm not sure how that got spelled that way. So the first one goes through the fundus, right? And once your food hits your stomach and we mix it up with hydrochloric acid, we don't call it food anymore. We call it chyme, CHYME. We'll talk about chyme a little bit more in a minute. Basically, that mixture is moved around by the rugae of our stomach. Those are the folds in our stomach, and it kind of just gets all mixed around in there. Once again, we're trying to coat it with this hydrochloric acid, and I'll talk about the function of hydrochloric acid here in a second. So first it passes into the fundus, then the corpus, then the antrum, and as it moves and is stored in the stomach, it's constantly being moved around and mixed with this hydrochloric acid, which has a couple of purposes, which are really important. So we'll get to them. The stomach is namely a storage tank, so that if we push things into our small intestines too quickly, we increase the rate of what we call motility and we don't absorb as quickly, right? So we want things to kind of go in a little bit of time into our intestines and move through pretty slowly so that we can absorb what we can. And that's kind of the job of the stomach. It just releases things into the small intestines a little bit at a time. And of course, it mixes up that food with hydrochloric acid, creating this thing called chyme. So these gastric juices are then kind of, the saliva from your mouth, the things that we've eaten, the liquids that we've eaten, all mixed together in this thing. Chyme is, as it's listed here, homogeneous. It's kind of all of same, kind of makes everything the same. It's acidy, it's soup-like, it's pasty, it's chyme. So the reason why these liters are here, and I wish the slides will actually pop those bottles up. The stomach has a normal capacity of 1.5 liters, so almost a full one of these, Coke bottles that are on your screen, so basically to the top of the label. But it can expand to hold up to 6 liters, which is all 3 of these combined. So you can imagine how big your stomach can get. We don't necessarily want to do it that way, but it does have that capacity. And to get 6 liters, you have to kind of train it. So it's probably not something you really want to do. The last bullet here says that the chyme passes then through the pyloric sphincter into the small intestine. So once it's all mixed up and mixed with chyme, hi Seneca. It's open. It enters into the small intestine. So the small intestine is called small because of the diameter, right? It's not length. It's all about the diameter. So small intestine is small because of the diameter. It's this inside one surrounded by the large intestine. It's 2 to 3 meters in length. It's only 3 to 5 centimeters in diameter, and it's made up of 3 sections that you'll want to remember: duodenum, jejunum, and ileum. About 95% of our absorption is going to take place here. And once again, if you think about the process, right, we've got chyme now moving into a small intestine. It's going to pass over what we call rugae. Kerckring. These folds, these Kerckring folds, are made up of villi. That's the little bumps you see down here where my cursor is. And those villi, those little bumps, right, are made up of microvilli and lots of little microvilli. So that's the fuzzy thing you see on top of the villi. And that fuzzy microvilli, that little blanket of them, is called the brush border. And it's through the brush border or these microvilli that we get absorption. So if you look at this one here where my cursor is, you're going to see the microvilli, right? That's the brush border. It's the entrance into the cell. There's a nucleus. If the nutrients then pass through the cell, and then on the other side of this cell would be your circulatory system. So your circulatory system is obviously made up of capillaries, which are the junction of an artery and a vein, and something called a lymphatic system. And in that small little space in that brush border, in that microvilli, is the lymphatic duct. Now, remember that, because that becomes really important when we talk about fat absorption. Most everything else is going to go into the circulatory system, and more accurately, a vein, because it's going to go back towards the heart to get, and it's going to go through the liver, but it's going to go back towards the heart. So it's going to get picked up by the vein. Okay, Kerckring are made up of villi. Villi are covered with brush border, and those brush border are little tiny microvilli. Okay, try to kind of wrap your mind around that. By doing this microvilli along the brush border, we increase the surface area of the small intestines by 20 times. So that's a lot of surface area to do some absorption. So like I said, the first 2 sections of your small intestine, 95% of the absorption is going to take place. So most everything is digested by the time it gets to the ileum of the small intestine, and then absorbed. All right, gallbladder. Your gallbladder stores bile, and bile is a salt which helps to emulsify fat. Now, if you ever wash dishes and you've washed something greasy, that's why you add soap. Soap is like the bile. Soap doesn't break down fat, and neither does bile, but what it does do is it allows water then to bind to the fat. It emulsifies it. It opens it up so that water can then have an effect on it. And I guess in a way that breaks it down, but it really does just allow that water to have an interaction with it. Now, bile that is stored in the gallbladder is made in the liver. So if you don't have a gallbladder, you can still secrete some bile, but it comes directly from the liver. There's no storage for it. So those of you who know somebody or don't have, or you yourself don't have a gallbladder, you can still eat fatty foods, right? But you can't eat a huge quantity of them because you don't have a gallbladder that can store enough bile to, because it's going to secrete enough bile to make to emulsify the amount of fat that you've put there. So it self-regulates basically. So that's what the gallbladder does. It's this little green sac right here up underneath the liver. Okay, it's this little green sac. Yes, this is where your gallstones can occur, right? So it's a bile salt, and it's because it's a salt, it can harden. If you have a healthy gallbladder, that's not going to happen so much. That gets secreted into the duodenum through the hepatic duct. So the duodenum, right, is the first part of that intestine. So just as it comes out of the stomach, the first thing it gets hit with is bile from the gallbladder. We're also going to get some stuff from the pancreas to take care of the rest of the things. So here we go. So the pancreas is located behind and below the stomach. You can kind of see a shadow of the stomach here, and there's your gallbladder. It looks like a corn cob, pointy corn cob. It secretes sodium bicarbonate, another form of sodium, and it's going to buffer the hydrochloric acid, right? So sodium bicarbonate may sound familiar to you. That's baking soda in another form. So basically baking soda buffers hydrochloric acid. We need to buffer it, right? Buffering means we're going to dilute it or neutralize it. And we have to do that because we don't want hydrochloric acid in our intestine. It doesn't mix well. It doesn't do well with that little brush border. So we're going to try to get rid of it. But we need the hydrochloric acid because the other things that the pancreas secretes will identify with what was already touched by hydrochloric acid, and then those enzymes break down protein. So we're going to look at that here just in a second. Just to give you an idea, we need that hydrochloric acid to begin to digest the food, but we also need it to signal enzymes to break down protein. So we need it. It's good stuff. Once it gets through the large intestine, that chyme then moves through this ileocecal valve and into the large intestines down here. So the cecum is the end of your small intestine, and then that's the valve that goes into the large intestine. Now, right next to that valve is the appendix, if it gives you an idea of where you're at on your large intestine. Then you have your ascending transverse, then it hits a downhill slope on the descending colon, sigmoid, and then out. So that's your large intestine. Now, what is it doing here? Well, basically, we're going to pull the water out of the chyme. And as the water comes out of the chyme, we create something called feces. So chyme becomes feces as we take the water out. So one of the main duties of the large intestine is to kind of reabsorb that water, take that water back out of the food that's in there that we've consumed or pushed through there with the stomach acids, so that we can use it in our body. So that's one of the main reasons. The other one is it can help with some more digestion. If we have any leftover sugars and fats, it can break down through the bacteria in our stomach. Will help break that down. We'll talk a little bit more about that here in a second. The colon, like I said, divided into ascending, transverse, descending, and sigmoid. If you want a picture of that, just right there, up, over, down, sigmoid. So up is ascending, over transverse, down sigmoid. Okay, so like I said, absorption of water. Then we're going to dry out that chyme and make it into feces. Once again, the longer it stays in that large intestine, the more water we can get out. And that large intestine is also influenced by that smooth muscle and peristalsis. So if we do anything like consume caffeine or any sort of stimulant, we smell more food, we increase the desire to eat, that'll move along that peristalsis faster, and therefore we don't pull all the water out of that, right, of that chyme. So that can make looser bowels, right? It loosens things up and they're not as tight. So it gives you an idea of why you have different feces at different times. Okay, the last part of this digestion, right, is digestion is breaking down something. So we're going to break down the food into chyme. The chyme then is broken down into its very smallest parts. And in the world of carbohydrates, that starts out as a large carbohydrate, right? And through pancreatic amylase, and now amylase with the ASE activates on amylose. That's this one up here. Must be amylose because amylase is the enzyme. And when we break up amylase, we're getting some maltase in this case. So maltase, or maltose. Maltose, the sugar, is broken down by maltase, the enzyme. All I'm doing is changing the O to an A, and then we know it's the enzyme. Maltase breaks it down into 2 glucoses. Actually 4, because there's 1, 2, 3, 4 here. And here's the 4th one down here, so don't get too confused. Goes through, this would be the brush border where my cursor is, in the cell and into the capillary. Protein is broken down by trypsin and into peptides, and then we use an enzyme called peptidase, and it breaks it down into amino acids. Those can then travel through absorption, when we'll talk about the absorption process in a second. Fat has big globules, and bile salts activate those, emulsifying the droplets. We get an enzyme that activates on those now emulsified droplets and breaking it into free fatty acids and monoglycerides, which can smoothly pass through that cell wall, mostly through osmosis, which we'll talk about in a second. But look where it goes, right? Lymphatic capillary, lymphatic duct. So that's going to go a different pathway to our heart than does the blood capillary. So keep that in mind. Carbohydrates, absorption. Okay, so we've digested it, right? There's the digestion. That's the last step of digestion. We break it down into its simplest forms. In this case, glucose for a carbohydrate, amino acids for proteins, and fatty acids and monoglycerides for fat. Once you get it down to that point, we can move it through the cell. We start breaking that down in the mouth. Amylase is then slowed down in the stomach because of the hydrochloric acid. So we're not getting as much breakdown to those simple forms. We're going to do more of a rough breakdown in the stomach. The duodenum gets some more amylase from the pancreas, right, to break down those starches. And then, like we talked about before, it moves through the intestine, and it's going to get to that cell wall on the brush border. So here it is, right? So here's the enzyme. In this case, lactose, the disaccharide, is broken into these 2 monosaccharides, glucose, galactose. Sucrase breaks down sucrose into glucose, fructose, right? And maltase breaks down into 2 glucoses. Now, if we're looking at these monosaccharides over here on the right, glucose is the common denominator for all of them, and our body loves glucose. Glucose is easily made into glycogen. Lactose takes a little longer of a process, fructose a longer process, and so we really like the galactose stuff. So when you look at products that are man-made for easy digestion and for like a triathlon or some sort of like gel that someone's going to take, it's a lot of maltose because we get 2 glucoses out of it. Just a little hint there. If you ever want to design a product or take a product that has really good energy source, glucose is what we're looking for. Okay, fiber is also a sugar. It's called cellulose. There's the OSE. But the thing is we don't have a digestive enzyme for it. There is no such thing in our body as cellulase, so we can't break it down. So fiber works really nice in our body to help clean out either our intestine. If it's water soluble, it can pass, or fat soluble, we can pass through the cell pretty good, and then we can use it to clean out our circulatory system. We can get some breakdown of fiber in our large intestine. So what's not absorbed can go into our large intestine, and the bacteria that can then break it down. Obviously, the byproduct of breaking down something in your large intestine through bacterial digestion is methane gas. So we're going to try to avoid that, right? We don't want gas formation. It's a natural thing, but it's uncomfortable. Okay, so fat digestion, starts in the mouth as well. We get a little linguinal lipase. There's the ASCs. We know it's an enzyme. It's going to break down some of those small triglycerides into fatty acids. So if the fat globule is pretty small and we can already get to it, we don't need to buffer it or emulsify it. We can break it down pretty quickly. Once again, it's formed in the stomach. We form chyme by its mixture with hydrochloric acid. And these are bigger fatty molecules, so it has to be emulsified by bile from the gallbladder in the duodenum of the small intestine. At that point, it's ready to be activated by other enzymes, and we can then absorb it through the brush border of the Kirk ring in the small intestine. This is what emulsification kind of looks like. So we have water, it's clear, obviously. The yellow is then the fat. And once you drop N-abyl salt, you can see that it's breaking the bond and mixing it down here with the water. So it basically is just adding soap to it, right? You think of it that way. Protein digestion doesn't take place in the mouth so much, mostly in the stomach and the small intestine. Proteases in the stomach acid do most of the work. And so basically the protease is an enzyme, but remember, all of our body is made up of some sort of protein, including our stomach. So we don't want free protease in our stomach, right? So hydrochloric acid keeps it from doing that. We have to buffer the hydrochloric acid, but the buffering process allows protease then to break it down in the small intestine, which is great news because we don't want a stomach ulcer. And basically a stomach ulcer is created from this protease that gets into our stomach. So we don't want that. Protease occurs in the small intestine after hydrochloric acid is buffered. Protease comes from the pancreas. Okay, here's that hydrochloric acid. It's strong. It's released into your stomach. It breaks down the food. It activates protease. It kills a ton of bacteria that we put in our mouth, right? So it kills a lot of it, but oftentimes we can consume so much bacteria that our hydrochloric acid can't buffer it all. And then it goes into our intestine and then we can get really sick. So our body recognizes that pretty quickly. That's why if we eat too much bacteria, like something bad for us, a rotten fruit or a rotten piece of meat or rotten cheese, we feel pretty nauseous fairly quickly. And then we'll throw it up. Our body has a mechanism, obviously, to get rid of some of that bacteria. Because hydrochloric acid does a great job, but we can't abuse it, right? We can only do so much. Okay, so once everything is broken down, we've got enzymes at play, we've made them into the smallest unit that we can make them, then we can absorb them. So digestion occurs first, and then we absorb. We can do it through either active transport or through simple diffusion. So active transport is a little interesting. It requires a little bit of energy, requires work, so therefore it requires energy. So basically you have a transport protein that has a certain shape to it. Okay, it looks like that, for example. And then I have a protein that floats by, or amino acid that floats by, that I want to get through the cell wall, okay? Or I have a molecule of glucose. And if it's glucose, then this little clippy thing is insulin. That gives you an idea. So basically it's going to grab it, right? If I was going in a muscle cell. So if not, it's typically through an ion channel. But let's say active transport, it grabs onto it, and then the shape now allows it to pass through the cell. So that's active transport. Simple diffusion is like osmosis, and it's a pressure gradient. So there's your active transport. It requires help through an enzyme. So I'm not an enzyme, sorry, a protein transporter. Or a molecule of salt or something to open up an ion channel, which we'll talk about here in a quick second. So here's simple diffusion, right? You have a pressure gradient on one side and a smaller pressure on the other, and it's just going to push through that membrane until both sides are equal, which is this bottom one. It's commonly known as osmosis. So there you go. Simple diffusion. It's just about pressure, right? You've got a big party going in your house and everybody's crammed in your kitchen. People start to move into the living room by just naturally just start sliding out because it's too crowded. So that's the same thing, right? The pressure is just too much. It starts to push through the membrane and into this more where there's more space. And in a perfect world, it's balanced. Wouldn't this be nice? Put your head on your books, and through osmosis, all of the knowledge from the books just leaks into your brain. It'd be awesome. Okay, this is what it looks like at the cellular level. This wavy line right here and down the middle is your microvilli, good, your brush border. Hope that you were saying that while I was saying it. Purple represents the carbohydrate. If we look at how it passes through, we have something called an active transport with sodium. Basically, sodium contacts the edge of the cell. It recognizes that it has glucose. Sodium ion channel opens up, the glucose floods in, and then it passes out of the cell fairly easy through facilitated diffusion. So some other markers will help pass it through the other way. That's glucose and galactose. Fructose goes through facilitated diffusion just like it passes out. So it just depends on the shape of the sugar, right? Now, all monosaccharides are C6H12O6, but depending on where the hydrogen and oxygen are on the ends of that carbon chain, will give it a different shape. Amino acids are kind of the same way. Once you get an amino acid that's simple, it's just one, active transport through sodium as well. And then you have these bigger amino acids. or peptides that have to go through with hydrogen. So it just depends on, once again, the shape. So the bigger they are, they have a little different shape. Hydrogen helps. If it's a simple amino acid, it's just sodium. This is way more than you need to know for the class, but it's kind of good knowledge to know that the more complicated foods that you eat require a different method of absorption. So that slows things down a little bit, right? So that's why when we say eat something that's a monosaccharide, glucose or galactose, because it moves through more quickly. So, if you're really getting down to the nitpicky business of digestion and absorption, that this is pretty important to know. Fatty acids are simple diffusion, in and out. So because the cell membrane is made up of fat, so in and out. As long as the molecule is fat enough, as long as the molecule of fat is small enough, it'll pass right through. Here's a diagram of what that looks like. So here's the purple and the green proteins. They get into the capillary. It flows in the blood. It goes through the hepatic portal of the liver, gets scrubbed out, cleaned up by the liver, and then into the heart through the inferior vena cava. The fat molecules then go into the lymphatic system through the lymphatic vessel. They bypass the liver. They go right into the left subclavian vein, which is above your heart, and drop into your superior vena cava, into your heart. Take home message here: fat molecules drop right into your heart. Everything else gets scrubbed by your liver. So the only thing that doesn't do that are the really, really small fatty acids. They travel the same line as proteins and carbs. But your bigger fat molecules, right, go through the lymphatic system and directly into your heart. That should tell you something. That's why we're trying to be careful on how much meat we eat. If we eat more high density lipoproteins, we're a little bit better off. That from your vegetables, for example, or legumes, things like that. Okay, water absorption, and 99% of water absorption takes place in the intestine. It gets passed through the cell through osmosis, just a pressure gradient. And then the vitamins will kind of do the same thing. Once again, with the vitamins, we only use pretty much what we need, and then the rest is expelled. So once we absorb it, it goes through the same process, and then our body picks up what it needs, and then it's flushed out through our kidneys. So that's why we have to keep taking vitamins, right? Every day. We have to keep our body with the availability of vitamins. The only way we can do that is to eat them. The only caveat to that maybe is calcium, which is made up, our bones are made up of that. So we can use calcium for that to release that. Calcium releases that ATP bound at actin-myosin. So without calcium, we cramp up, right? So when we get cramps on the football field or get cramps in athletics, we tend to give people salt. Well, all that's doing is opening up a channel to let calcium flood in and release that contraction. So we take salts to let the calcium in. Calcium is actually the thing releasing the cramp. So that's the only mineral probably that gets stored in any great abundance in our body. And we don't really want to leach it from our bones, right? So make sure that we're taking our vitamins and minerals pretty regularly so that we can use it, because absorption is going to occur, but we only use what we need. Same thing with the minerals. Just talked about that. So when I already talked about bacteria in the colon as well, there is some production of vitamin K, B12, some other B vitamins, riboflavin, thiamine. So we have about 100 million of these, and we can put more in by eating well. Yogurt is a good source of some of this good gut bacteria. So, all right. Some gastric emptying. The reason I show this slide is because oftentimes in athletics, we're nervous already, and just some of these bullets will cause our peristalsis to speed up, and we really don't want that right before an athletic event. So the smaller side of food, thinking of food, how much we drink, especially if it's salty or sugary. So there goes to the energy density. So that sugar density of the drink. Anything about 8% or more per volume is sugar, then you're going to speed up that peristalsis. So you want to drink energy drinks or Gatorades that have less than 8% per volume. So your can should say how much is in there. It'll give you a percentage. So less than 8, more like 6. If you can get it less than 6, that'd be great. So Take a look at your Gatorade bottle and make sure it should say 6%. The Gatorade used to be 8%, but they've reduced it because of this. So that was years and years ago. Temperatures, warmer the better. Or the warmer, the more, the quicker the motility, the more peristalsis. So you want it kind of tepid, roomish temperature. Exercise tends to slow down, right? And your peristalsis and high intensity. Women have a slower rate of motility or peristalsis than men. And of course, if you're nervous or stressed, you increase peristalsis, so that while your stomach really feels like it's got butterflies in it, or you feel like you have to go to the bathroom, that will signal your body to pick up your peristalsis. Okay, there's your terms. Hope this helped a little bit. I know it was fairly quick, but you should have looked through the slides and then do this. That's usually the best way. Questions, just please text or email me, and I'll be glad to answer them. Okay, keep up the good work.